Welcome, everyone. We're pleased and honored to be hosting Dr. Akiko Ivasaki this evening and to have so many people in attendance. I'm Leslie Krumholtz. I'm the co-founder of Hugo Health Kindred. And for those of you that don't know, really quickly, Kindred's building a network of what we call data-enabled people who've been impacted by COVID and who want to contribute to research in partnership with leading scientists. I strongly encourage anyone who is not a member of Kindred to check us out. I have put a link to the website as well as information about the Listen study. If you're interested in joining Listen, you need to actually join Kindred first. The Listen study is being led by Dr. Akiko Ivasaki and Dr. Harlan Krumholtz. So I'm not going to take up another second of your time. I'm going to pass this right over to Akiko for introductions, kick off this very important presentation. And once again, just to say thank you so much for spending an hour of your evening with us. So Akiko.
Thank you so much, Leslie. I'm delighted to be back on the town hall again with the Kindred Hugo Health. And I'm also delighted to be sharing the stage with my colleagues, Ornali, Cesar, Harlan. So very happy to be here. All right, so I'm going to jump in to my talk. Let's see. Okay, hope you can see this. Okay, so what I wanted to do today was to give you an overview of our current understanding of long COVID, particularly immune responses in long COVID. And also, I'll spend some time at the end speculating on how vaccine-related long haul could occur based on some of the data that the Yale Listen study has already collected from many of you participating tonight. So before I go into discuss long COVID, I just want to emphasize that COVID is not the only infection that results in post-acute infection syndromes. And I was fortunate enough to co-author a review with Jan Chotka and others on this topic, which is really important to keep in mind because it means that many other viral and non-viral infections can lead to prolonged symptoms, some of them leading for decades of disease and others kind of having an onset that is much later than what we're seeing with long COVID, something that happens decades after the infection. So there's a lot of complexity into these post-acute infection syndromes. They haven't been really well studied. And that's something that we're going to change by studying these diseases at the molecular and immunological level to understand what might be going on. So the long COVID pathogenesis, there are multiple hypotheses that have been raised. I just want to go over the four major ones. There are many others that have been proposed. The first hypothesis is the viral reservoir of viral pathogen-associated molecular patterns. And this is, hypothesis that says that even though the viruses are considered acute infection, it's possible that these viruses may remain in some form of replication capacity, may not be infectious particles, but they are remnants or they are parts of the virus that resist being removed. And that could be persisting in a person. And that could lead to recognition of these pathogen-associated molecular patterns like RNA structures, or it could also lead to the expression and persistence of viral antigens that lead to chronic stimulation of lymphocytes. So that's one hypothesis. Another one is autoimmunity. Many infections precede the onset of multiple types of autoimmune diseases, multiple sclerosis and lupus and many others. So it's possible that the COVID infection can be leading to stimulation of bystander or molecular mimicry autoimmune responses, and that could be prolonged and leading to symptoms. The other possibility is dysbiosis of gut microbiome or reactivation of latent viruses. So all of us, many of us, most of us carry multiple different viruses, and many of these viruses don't cause any diseases, but they are remain in the host as a latent form. And these types of viruses can become reactivated upon immunological stimulation or immune suppression that may be caused by COVID. And that reactivation itself could trigger this virus to become activated, replicate, and then cause disease. The final possibility is tissue damage. And this is, virus infection and or immune responses that are induced by the infection that can be triggering tissue damage that is hard to repair, like fibrosis. And that could be lingering or very difficult to restore in a long term, and could be leading to disease. And I'll give you examples of each of these hypotheses. So there are over 100 papers now demonstrating some form of viral antigen or RNA that's present in people with COVID months after the infection. And many of these virus antigens and RNA has been located in the gastrointestinal tract. So this may be a place of reservoir, or at least some antigen being remnant there. There was a very nice study by Jim Heath's group that demonstrated that EBV, Epstein-Barr virus viremia at the time of COVID diagnosis is one of the 4 predictive factors for developing long COVID over the 3 months period that they were studying. So it's been reported in other studies also that EBV can reactivate and seems to be happening preferentially in people who develop long COVID. The other finding from the same paper demonstrated that lupus-related autoantibodies are elevated at the subclinical level in patients at the COVID acute phase who then go on to develop long COVID, the second of the 4 predictive factors for developing long COVID. And then there's this tissue damage. So we, with Professor Michel Monge's group at Stanford, demonstrated that even a mild respiratory only infection with SARS-CoV-2 in the mouse model can lead to significant damage in the brain for extended time period for over 6 weeks post-infection, whereas similar types of prolonged damage was not seen with mild influenza infection. So there's something about SARS-CoV-2 that may trigger this long-term tissue damage, even if the infection itself is completely resolved. So we suspect that long COVID is multiple diseases under one umbrella, and that there are multiple endotypes of diseases with different drivers, molecular drivers. And if we can subset long COVID into the right types of disease drivers and target the root cause of disease, that would be the best way to go forward with therapeutics. There's also the interesting paper where they train dogs to detect inactivated SARS-CoV-2 infected supernatant. And these dogs were able to identify in a blinded manner 51% of the long COVID patients, their clothes, whereas 0% of the controls were identified by these dogs. This, to me, indicates that there are volatile organic compounds released by the people with long COVID and likely having to do with the virus infection itself. That's why I put that in the viral reservoir category. And there's also papers from David Walt's group that demonstrated circulating spike protein in people with long COVID. So this sort of reservoir antigen RNA, there's a lot of accumulating evidence for it, but that may not be the only root cause for long COVID. So today I want to discuss our latest work on collaboration research that we've done with Mount Sinai group led by Dr. David Petrino, who is just an amazing human being and a great leader who is treating people with long COVID, thousands of people with long COVID from the very beginning of the pandemic. And his team, listed here, Jamie Wood, Laura Tabakov, Dana McCarthy, and our team at Yale have collaborated to dissect the immunological phenotypes of people with long COVID. And these are the co-first authors of the paper, John Klein, who led this study and currently working on the revision of this paper, along with Jill Jaycox, who is a MD PhD student in Aaron Ring's lab, who's using this rapid extracellular antigen profiling to look for antibodies against our own antigens, autoantigens, as well as viral antigens. Rahul is a brilliant student in David Van Dyke's lab who does machine learning on all the parameters that we've measured to try to predict which factors are most contributing to long COVID. Pei-Wen Liu tirelessly looks at everyone's peripheral blood mononuclear cells using real-time flow cytometry to look at the cell types in the blood. Jeff and Sasha have been working very closely together to look at the patient data as well as antibody reactivity to extra SARS-CoV-2 antigens. So this Mount Sinai Yale long COVID study is currently in medRxiv. Anyone interested can read this, but just to briefly go over, what we've done was to recruit participants from the Mount Sinai long COVID clinic and to study a variety of electronic medical records, symptom survey, flow cytometry to look at cells that are in the blood, human exoproteome. This is the REAP technology developed by Dr. Ring's lab to look at autoantibodies. We did SARS-CoV-2 antibody profiling, peptide display library to look at linear epitope mapping of antibodies from these patients, and used plasma proteomics to look at plasma factors that are distinct in long COVID patients. First, just briefly about the demographic nature of these participants. The long COVID patients listed always in purple have, most of them are between 30 to 60 years of age. There are some younger and some older. And similarly, the convalescent control, these convalescent control groups were people who were infected around the same time as those people with long COVID, but have recovered from COVID. And their age group was similarly enriched in 30 to 60 years of age, some younger and some older. There's no significant difference in the age, and the sex is female dominant. This is seen over and over in every study on long COVID. There's definitely sex bias towards female. Also, we focused on people who who were not hospitalized. We wanted to focus on so-called mild COVID that then turn into long COVID, because this is, we believe, is a distinct disease from those people who were in the ICU and who were receiving drastic medical treatment versus people who were staying at home and fighting the virus infection, but then develop long COVID. So days from acute COVID in the convalescent control and the long COVID are again not significantly different. They were overall 400 days out from the original infection. So these are the first wave of COVID that hit New York City. And so we're looking at a much later time point than most studies that have been reported. First, looking at the immune cells that are in the peripheral blood and looking at various different cell types, what we noticed was that there is increase in non-conventional monocytes. These monocytes are known to patrol the body for viral infections and other infections, so that's elevated in long COVID. There's a reduction in dendritic cell subset known as CDC1s, and these cells are critical in priming cytotoxic T cells and type 1 Th1 cells, which are important to fighting the virus infection. We also see elevated activation of activated B cells as well as double negative B cells. So these are the features that were elevated in long COVID. So it again suggests that the B cells are being stimulated by something, whether it's SARS-CoV-2 or some other antigens, we don't know. In terms of the T cell subset, so here we're listing the CD4 T cells on the top and CD8 T cells on the bottom. There's really no need to go into the detail of these markers, but suffice to say that the CD4 T cell and CD8 T cells, there is a significant increase in the exhausted T cells. And so these exhausted T cells have been seen in chronic viral infections and cancer when the T cells are stimulated over and over, seeing the same antigen, and they basically go into this state of exhaustion where they're no longer very functional. And so that is elevated in the long COVID patients. And interestingly, when Pei Wen looked at the ability of these T cells to secrete cytokines, so cytokines are important factors that are released by T cells to communicate with other cell types throughout the body. What we saw was an elevated cytokine secretion or production from CD4 T cells from long COVID patients for IL-2, IL-4, IL-6. These are cytokines, IL-4 in particular, are known as Th2 cytokines. And these cytokines are very important for fighting Helminth's infection, like these worm infections, but are not very effective in fighting virus infections. And we're seeing elevated levels of all of these cytokines, in particular, the IL-4, IL-6 double positive T cells that secrete both of these cytokines, were pretty much only uniquely found in the long COVID patients. So this is interesting because the kind of T cell you want to fight a virus infection is known as type 1 or Th1 immunity. And we're not seeing that. We're seeing something that's diverting from that protective immune response against viruses. We then looked at antibody responses to SARS-CoV-2 antigens. So what we noticed was that antibody against the spike protein or the S1 region of the spike protein or the receptor binding domain that actually binds to the target cells, they were elevated in the long COVID patients over healthy controls or convalescent controls. So the healthy controls, I forgot to mention, are people who've never been infected with COVID, but live in the same area. And all of these people have gotten 2 doses of mRNA vaccines. So they should have equal levels of antibody. And yet what we're seeing is an elevated levels of anti-spike antibody from long haulers. And I'll come back to the functionality of these antibodies later. We next looked at what are the most distinct factors that are found in long COVID patients compared to non-long COVID patients or controls. And what we found was that the most significantly different factor we found in the plasma of long COVID patients were the cortisol level. So cortisol is a very important hormone. It's known as stress hormone, but that is a bit of a misnomer because it's needed for everyday physiological function, like how we nutrient handling, glucose utilization, wakefulness, many different aspects of physiology is controlled by cortisol. And if you look at this panel here on the bottom, the long COVID patients in purple, they had about half the level of cortisol compared to the healthy control. And this is the most significant because they were uniformly lower than the control groups. Whereas other factors, there is a huge variation. Some people have very high levels and low levels, but the cortisol level was the most tightly different, distinct between these groups. So cortisol is secreted by the adrenal glands, and it performs very important function. It's a diurnal hormone, which means it is the highest level right around the time you wake up, and then it levels down during the day. And the wakefulness is controlled by cortisol. If you have very low cortisol, it's very difficult to wake up and be motivated to start your day. And it's tightly regulated through this hypothalamic, pituitary, adrenal axis, the HPA axis. And so we wondered this low cortisol, whether it's driving higher levels of this other hormones from the pituitary called ACTH. Usually when you have lower than normal level of cortisol, ACTH goes up in order to compensate for that low level. So you make more and more cortisol. That is not what's happening with long COVID patients, meaning that something upstream of adrenal gland, potentially the pituitary glands or the hypothalamus, there may be dysfunction up in the CNS region, and that's leading to this lower levels of cortisol. And it's important to note that the collection of the plasma or the blood was around the same time in all the three different groups, meaning that the changes in the cortisol level has nothing to do with the diurnal nature or when we pick these blood samples from these patients. So, and then cortisol is number one, but there are other inflammatory factors such as interleukin-8. IL-8 is a very well-known chemokine that attracts neutrophils. These are highly inflammatory cells. We also have many different chemokines. Again, chemokines are these factors that recruit white blood cells to many different tissues, as well as some complement factors. So there's definitely something inflammatory going on. What's driving this and why the cortisol level is lower is currently unknown. We suspect it's something to do with the HPA axis deficiency. Okay, so what about these other hypotheses? We didn't look at dysbiosis in this study. We probably should do this in the future, but we did look at reactivation of latent viruses. And we did this in 3 different ways and found the same answer. So one way in which we did this was to look at the REAP score. This is the rapid extracellular antigen profiling, which enables us to look at antibody reactivity to like thousands of different antigens, some of which were viral antigens. And as I mentioned just a few slides ago, the antibody level against the spike receptor binding domain was elevated for long COVID patients. However, what was striking is that we also saw elevated antibody levels against these latent viruses that normally we wouldn't have these antibodies against. So Epstein-Barr virus, varicella zoster virus, these are sort of mononucleosis and chickenpox viruses that most of us, before the vaccination for chickenpox, most of us carry these viruses inside of us without any symptom. But under certain stress, such as a COVID infection, some people are elevating this level of reactivation of these viruses. And what's intriguing is that this is highly elevated in long COVID compared to the convalescent control or the healthy control. And what's also important to note is that the seroprevalence, meaning that how many people have latent viruses, there was no difference in these groups. So it's only the reactivation dependent antibodies that are elevated. And there were some other differences, like herpes simplex virus specific antibody was slightly lower than the controls. So I told you we did this 3 different ways. Another way we measured EBV reactive antibody is through Serimmune, which is a linear epitope mapping strategy, very different from REAP, but we found the same answer. So this is the REAP data for the GP42, which is a glycoprotein on the surface of the EBV. The IgG against this is elevated in long COVID. And also this middle panel is from the Serimmune analysis. We found not only GP42 reactive antibody elevated, but we can pinpoint the specific amino acid sequences within the GP42 that the patients are reacting to. And we mapped it right here in this pink. So this particular set of amino acid is being recognized much higher in long COVID patients compared to the controls. And GP42 is a very important viral antigen that is required for entry into B cells. So activated B cells, GP42 reactive antibodies, type 2 is like IL-4, IL-6 secreting CD4 T cells. They're coming together to tell us something. And indeed, when we look at the correlation between GP23 or 42 specific antibody and IL-4, IL-6 double positive CD4 T cell frequency, we see there's a linear correlation in long COVID patients, meaning that these things may be linked to each other. And it's well known that EBV, because they express this GP42, blocks the T cell activation through blocking of MHC class 2. And they kind of divert these T cells into a Th2 IL-4 secreting cell type compared to Th1. So in our heads, we're kind of making some links in between these observations. What about autoimmunity? Well, we found a lot of functional autoantibodies during severe acute COVID. When we looked during the early phase of the pandemic, hospitalized patients and ICU patients had a lot of these autoantibodies that block the immune system itself, like type 1 interferon specific antibodies, for example. So we were expecting to see a lot of autoantibodies, but we didn't. We did not see autoantibodies significantly different in long COVID patients compared to controls. We did this in many different ways. This is the Jill Jacobs work with Aaron Ring, and this is basically looking at different patients on the different columns, and different rows indicate autoantibody presence against different antigens. There really isn't a universal pattern or anything enriched that we can see in the long COVID patients compared to the controls. And antibody reactivity per patient was not different. There was no difference in antibody reactivity number to long COVID propensity score. This is sort of the severity score that we calculated. So all in all, we don't see a signature for autoantibodies against extracellular antigens. It's possible, though, that there are intracellular antigens that are being targeted by autoantibodies, and that's something that we need to separately examine. Using the machine learning algorithms that have been conducted by Rahul in David Van Dyke's lab, we were able to separate long COVID versus non-long COVID participants just based on immunological phenotyping alone, with a 96% accuracy. And when they asked what are the factors contributing to this distinction of long COVID patients, they saw that autoantibody had very little predictive ability, whereas antibody against the spike of SARS-CoV-2 had some predictive ability, but it was really the cytokines flow cytometry and antibody against EBV and things like that that were able to distinguish people with long COVID versus those without. And when they looked at all the different parameters that we've included in the analyses and asked what are the most significant and most differential factors that we see that are either lower in long COVID or higher in long COVID, we found that cortisol came out as the number one factor, as a lowest factor in long COVID patients with the highest degree of specificity. But there were other things like the TCM, these are the central memory CD4 T cells that were also lower. And then CD8 T cells, for example, were also lower, but not as significant. What's higher in long COVID patients are these activated B cells, EBV reactive antibodies, and exhausted T cells. So this is starting to paint a picture of dysfunctional immune responses and reactivation of endogenous viruses that may be sort of distinguishing at least the biological factor for long COVID. So these are the key findings from this study. I didn't have even time to talk about patient-reported outcomes, but they alone were able to predict or identify COVID patients with 94% accuracy. So ask the patients in order to diagnose. Second, immunophenotyping revealed these distinct increases and decreases in different cell types, I guess notably the exhausted T cells being elevated. SARS-CoV-2 specific antibody response, particularly spike-specific ones, were elevated. Evidence of herpes virus reactivation, EBV and VZV, were detected in a subset of patients. We did not see any significant increases in autoantibody to extracellular antigens. And long COVID alone, using machine learning, can efficiently predict, immunological data alone can predict long COVID patients with very high accuracy. And the low cortisol level was the strongest predictor for long COVID. So I'm going to show you a couple of slides looking at sex differences in long COVID. This is not even on medRxiv yet. It's fresh off the press or whatever lab. And this is analyses that are done by Julio Silva and Takehiro Takahashi. They took the same set of my long COVID data and started to look at sex differences. And what's striking about this analysis is that female and male patients suffer from distinct symptoms. There are some that are overlapping, but some that are very distinct. So females are indicated in blue and males are in pink. So please forget your stereotype. Basically, over here on the top, with the curves are overlapping with each other, there are sort of the common symptoms that is, for example, sleep, disorientation, urinary issues, ingestion reflux. These are similarly reported for male and female patients. Whereas there are some symptoms that are slightly higher in female over male, but they're not that separated. Whereas there are these sets of symptoms, at least in the my long COVID participants, were quite different. There were female dominant symptoms that included numbness, dizziness, and many other features that are listed here. And there was only one that was significantly male dominant, which is sexual dysfunction. And hair loss is the most significantly different reported symptoms that were predominantly female. And if you look at the symptom burden, there is overall higher symptom burden in the female. And then organ system involvement was also higher in the female participants. And what was another striking thing that we found was that in long haulers, especially in females, I already told you that long haulers have higher levels of anti-spike antibodies. But when you ask the question of, are these antibodies functional in neutralizing the antibody, the answer is no, that these people with long COVID, even though they have elevated levels of anti-spike antibody, their functionality is quite low. So female and male, if you look at the long COVID compared to control, the steepness of the curve indicates how potent the antibody is against neutralizing the virus. And you see that there's a lot lower slope for the female and male participants. And when you calculate the relative potency, the female long COVID patients have the lowest level of potency with respect to neutralizing antibodies compared to male patients. So even though the elevating levels of antibodies suggest that there's persistent antigen, we are seeing that these antibodies are not very functional. So it would be consistent with presence of virus or antigen remaining in these people, but these are pure speculation at this time. And people with high disease burden had tended to have lower levels of antibody potency compared to those with moderate levels or the organ system involvement. Again, it's consistent with the notion that if you have very high potency antibodies, you're slightly better off with the symptom burden. So based on these hypotheses, data, we hypothesize that the original 4 different pathways to long COVID, it's unlikely, or at least for the extracellular antibodies, we don't see much correlation of autoantibodies for long COVID. And we also think that with the increased levels of antibody with decreased level of potency in long COVID, it's possible that it supports this viral reservoir hypothesis. And we also found EBV and VZV reactivation that is much more prevalent in long COVID patients. So, and then tissue damage we did not look at, we just looked at the blood from participants. So we don't know the tissue level analysis yet. So this is sort of like what we're seeing overall. It's still a very, very early phase. This is a sort of hypothesis generating research. We need to do a lot more work to understand this better, but at least we're seeing some hypothesis that's more likely or less likely. And what I wanted to do for the remaining, hopefully 5 minutes or so, I don't know if I can do it so quickly, but I wanted to share with you a, again, unpublished data on post-vaccine long haul. So this is all based on Yale Listen study that many of you are participating. And this was done by Bornali, who is on the panel tonight. So any specific question you can address to her. But she asked the question, how do symptoms and demographics compare between long COVID and post-vaccine long haul? And because the Yale Listen study already has many participants who have either long COVID alone, the purple is the PASC control, PASC participants, and green bars indicate vaccine adverse events. The grays are people with both, who have both long COVID and post-vaccine long haul. And here's the demographics. There seems to be, again, dominant female over male, the numbers of participants reporting these symptoms in our study. And then total number of participants in these 3 groups is 206, 135, and 108. We would love to increase these numbers by recruiting more people into the study. This is already revealing something very important. So I encourage, for those of you who are not on the listen yet, please get on to this study. It's you basically join through Kindred online. And Leslie can help you if you have any problems getting on. And the mean age, again, is very similar, much younger than what you would see for severe COVID, but typical of long COVID. So Bornali wanted to compare the health status of people with PASC versus vaccine adverse event versus both. And these are your data, people who are reporting poor health, fair, good, very good, excellent, do not know. The percentages are very similar overall in the 3 groups. And also another survey with EQVAS, again, very similar pattern we're seeing. There's nothing significantly different in the 3 groups. This is based on a questionnaire that we have on Yale Listen. It asks you, have you ever been told by a doctor before COVID, so before January 2020, that you have any of the following diseases? And there are 2 questions here. But essentially, if you look at the bars, there are strikingly similar percentages of people reporting allergies or arthritis, asthma, whatever diseases you look at, they're very, very similar. There may be some differences in some of these psychiatric diseases, but these numbers are just very low. We can't really make any conclusions from this. We need more people to participate so we can understand the differences, if there is any. Another questionnaire, currently, have you ever been told by a doctor that you have any of the following? Again, there are two sets of questions. And of course, the most different answers turned out to be PASC itself. So people with PASC or people who have both, obviously they have the highest level of answered yes compared to vaccine adverse events. Whereas the reverse is true, people who have vaccine adverse events are reporting, obviously, these two last groups, but the PASC people, there's nothing. So this is sort of an internal control. The survey is working very well. But if you look across the other things, there were just very little difference. The only significance that we saw is migraines and neurological conditions that are slightly different between these groups. But again, we need a lot more people to participate so we can really understand, how significant these differences, if any, are. But overall, again, incredibly similar, right? This is a question about select all that you believe you have had as a result of PASC or vaccine adverse events. And this is a patient's own assessment of what symptoms may be attributable to long COVID or post-vaccine long haul. And again, they're very, very similar. There are some things that are slightly different, brain fog, memory issues, difficulty speaking, and so on. But overall, very similar. Now, I mentioned over and over, we need more people so that we can really look at this in a larger population. But so far, these surveys are showing us that these three groups are very strikingly similar. So currently, there's this remarkable overlap in symptoms and sex ratio for long COVID and age group. So based on this, what we hypothesize is that there must be a significant overlap in the drivers of these diseases. For instance, it's possible that the vaccine stimulation of innate immune responses could be similar between the acute COVID infection and vaccine, and what's triggered downstream is similar. And we know for both of these that there is inflammasome activation on RNA sensor stimulation. These are two key innate recognition pathways that are triggered by vaccine and virus. So that could be the commonality, and this should result in acute, within hours or days symptoms. Whereas vaccine stimulation of adaptive immune response, there's also one thing that overlaps between COVID infection and vaccination, which is the spike. So it's possible that anti-spike antibodies that form, is causing immune complexes that may be leading to vascular activation, microclots, or other issues. Now, whatever hypothesis here is absolutely speculative. There's no evidence for this, so please take this with a huge grain of salt. But based on the symptom data and demographic data, these are the kinds of things that we're starting to think about because of the similarity. There's also anti-spike antibodies or T cells that could attack spike-expressing host cells. It could be endothelium, it could be epithelium. Epithelium is definitely a target of virus infection. The vaccine can be taken up by different cell types and express the spike protein. So again, there may be some overlap on the vulnerable cell types. There may also be antibodies that target autoantigen that is similarly induced by spike in the virus, similarly for T cells. And vaccine induces potentially reactivation of latent viruses, in which case we're kind of triggering the same pathway as what we see with long COVID. So, oh yeah, I don't know if I have time for this, but just very briefly, I know some people are very interested in the peripheral pain and small fiber neuropathy. And I've been thinking a lot about this. There may be a way of connecting the low cortisol level that we see in the long haulers to release of immune suppression in the local tissue that enables sort of chronic pain trigger by damaging the endothelial cells within the skin, as well as the neurons themselves. And there are lots of regulatory and stimulatory lymphocytes that are controlled by cortisol that could be released as a result of this low level cortisol. There is also local factors that immune cells can secrete that can increase pain sensitivity or increase the pain-inducing factors themselves. So there's a lot to think about, but I'm trying to kind of put a lot of the findings that we've already have into trying to explain what may be going on. These are all just hypothesis generating things. And so we haven't tested these. Okay, I'm going to end here because I really want to take questions, but I can't end without thanking everyone who's like really dedicated to try to understand long COVID and post-vaccine long haul. And that includes not only members of my own laboratory, but the Yale Listen and Yale Recover study participants. Particularly want to highlight Harlan, without whom none of these Yale Listen study could be done. And also David Petrino, just an amazing partner that enabled this first study on my long COVID to be happening. And all the amazing people who do this immune profiling data analysis day in, day out, without whom none of these insights could have been developed. We're also expanding this to ME CFS cohort with the help of Amy and Michael and the PolyBio team, and also the CNS inflammation collaborators that I didn't really even have a chance to talk about today, but we are enabling these studies with a huge team of people who are dedicated to understanding these diseases. So thank you for your attention. I'm happy to take questions.
And I will start with the first question that's there.
Okay.
A young man told me that he had long COVID that caused cardiac symptoms. He said that he was sure it wasn't a vaccine reaction because his doctor told him he had long COVID. How would his doctor disaggregate COVID from vaccine reaction?
Yeah, that's very difficult to disaggregate because it's known that COVID can cause significant heart problems. I mean, Harlan is the expert cardiologist on this panel, but COVID itself can have a significant, cardiovascular problem, and so does mRNA vaccine in a subset of people, younger male, adolescent males, in a very small subset. So if he had COVID and have been vaccinated, it's a little difficult to tell what caused what. But if you look at the post-vaccine myocarditis studies, they often tend to occur a few days after the second dose. So you might be able to kind of look at the timing of the cardiac issues that this person had to try to link what may have led to this. Again, it's very hard to parse these apart.
The next question is, I would like to know if there is a study forthcoming in children living with long COVID. There are doctors at Yale Pediatrics who have been working on long COVID kids. I'm hoping that your team is working with pediatrics to start some longitudinal studies.
Absolutely, great question. So we're very fortunate to have one of the clinical fellows from Yale Pediatrics working with us on long COVID. So she and her colleagues are starting to collect biospecimen to be able to do similar kinds of studies. That study is just beginning, so it's not, we don't have any data or anything yet, but we would like to extend the study, of course, to children. Yeah.
Thank you, Akiko. The next question is, so somebody says, kudos to you, but however elegant the study, academic recognition of the pathogenicity of SARS-CoV-2 S protein via vaccine is impaired. All 5 of my physicians and myself, who is also a physician, were not aware of the clinical relevance of S protein vaccine in my significant CNS, autonomic, retinal, and cardiac events. There are publications for long COVID yet, which are silent about vaccine impacts. And this person is also talking about multiple recent articles. impacting spike in blood pressure dysregulation. Could you please comment on this?
Yeah, I mean, we need to change that. That's why we're doing this Yale Listen study, is we are going to approach vaccine related long haul with the same scientific rigor as we do for long COVID. And so hopefully this question can be no longer relevant, you know, in once we do these studies, and hopefully others are also engaging in similar studies. The vaccine-related adverse events are a very tricky issue that many people shy away from. Of course, all of us here are pro-vaccine people. I mean, I developed my own vaccines in the lab using a nasal spray. So we know the importance and the clinical benefit of vaccination, but we also have to acknowledge that there are a small subset of people, we don't know how small, but a subset of people who are suffering extreme, you know, health issues after vaccination. And it's not healthy to ignore or dismiss the potential link between the timing of the vaccination and the onset of these diseases. And if we don't look, we won't find it. So I agree with this person's comment that we have very little evidence for vaccine-induced impact on the health, but that's because we haven't looked. I think we really need to start looking at this. And that's the whole point of this comparison in Yale Listen study.
So the next question is talking about if we have looked at reactivation of bacteria like Borrelia or Bartonella.
Yeah, so the great thing about using this Sterimmune technology is that they have many tick-borne diseases and vector-borne diseases in their panel that's been verified using distinct cohorts that are prior to COVID. And so we are very fortunate to be working with their development team to be looking at antibodies against other pathogens that are not necessarily just viral, in order to be able to detect antibacterial, antiparasitic, antifungal antibodies that may be also elevated in post-vaccine or post-COVID diseases. So we are absolutely going to be looking at all of those.
Thank you, Akiko. The next question is, most general physicians have no information or ability on testing cytokine profiles. Are there any specific resources available to individuals for cytokine testing?
Yeah, there is a clinical test that looks at different inflammatory cytokines, but the types of cytokines and chemokines that we're detecting goes much wider than the clinical panel that the physicians normally use. And if you use those panels, you may not pick up the differences. So unfortunately, I don't know of any commercially available sites that would do a cytokine profiling for patients, but we are doing it. So if you are enrolling in our study, we will be looking at those, and we will be sharing some data back with the patients. Not everything, because some of them are very complicated in terms of interpreting the data, but for some standard things, we can certainly give back to the patients. And that's the whole point of this, collaboration with the patients. So yeah, if you're interested, again, join the study and we can work together.
So the next question is, was the reactivated virus, like were the antibodies IgGs or IgMs? Was any Lyme reactivation studied or seen? Yeah,
so we focus on IgG for now. We are doing some IgM and other isotypes just to be thorough in our analyses. We also did EBV viremia analysis, which is detecting the DNA from EBV in the circulation, and we don't see that. So it suggests that the reactivation must have occurred recently, but it's not an active viremia that's going on. And that's been seen by other groups as well. So it's the acute phase of the COVID that you're likely reactivating, but after that, the virus itself, the genome is gone, but you still have this antibody signature in these people.
So the next question is talking about cortisol. Do the lower levels of cortisol fall outside normal lab values for the time of day that they are being drawn in long COVID patients?
Yeah, so I should emphasize that all of our studies are done in the research labs. These are not the clinical cortisol measurements that doctors order. So, and we are actually trying to do a more thorough study on cortisol by collecting saliva from participants over 2 days. So many times sampling so that we can look at the diurnal pattern of cortisol level in long COVID participants compared to convalescent control and healthy control. So that study is ongoing. And once we have data from that's with my long COVID study. Once we have the data, we should be able to tell much better what is the pattern during the day, throughout the day, of the cortisol in the long COVID participants.
So here's a question which was sent by mail by one of the participants, I believe. I suffer from vaccine-related adverse events. Is there any test that can ascertain, if any, and how many spike proteins are in your system? And if so, is there any approach to remove them?
Right. So I did mention David Walt's paper that looks at circulating spike and nucleocapsid using a very sensitive assay called Simoa assay. And in their hands, they did see spike that's being elevated, especially the full-length spike, not so much the S1 region of the spike, but they were able to detect the full-length spike, as well as some nucleocapsid, but not too much. So it's really the full-length spike that's being detected more from the long COVID participants in that study. We have done some ELISA, which is another way of looking at proteins in the blood, and have seen some spike circulating in a subset of long COVID patients. There is no clinical test that doctors can order yet to look at these circulating spike. There should be, somebody should be developing these things, but we don't have it yet. And we're trying to develop our own way of analyzing, but it's not widely available yet. Okay, so the spike, is it the spike the entire problem? If we get rid of spike, do we cure people? We don't know. There are things that we could do to look at these issues in a clinical trial, but no one has done those studies yet. So for instance, we could imagine monoclonal antibody therapy to remove spike from the patient if it works well in various tissues. There are some areas of the body that's not accessible by antibodies, so we may need to design a much smaller molecule to get rid of spike. But again, we don't know whether spike itself is causing the disease or it's something that was triggered by the spike that continues to cause problems.
So the next question is from a participant who is in another part of the world, so is unable to join us. But the question is regarding the 2 hypotheses out of the 4 that you've discussed. So, so she is saying that in the previous meeting you had mentioned, like in today's meeting, you had mentioned that IL-2, IL-4, IL-6 cytokines are higher in long COVID participants, which means that a strategy could be to take ibuprofen, but if there is any presence of residual virus, that would not be a good thing to do. So she would like to hear your comment.
Yeah, that's the thing. We can't just, jump to a conclusion that these cytokines are just generally bad. So for instance, cytokine suppressing therapies may make things worse if these cytokines are actually keeping the virus at bay. And so, yeah, it's really difficult to recommend a particular therapy at this point because we just don't know enough. One thing that could be done is to treat the reservoir virus, if there is, with something like Paxlovid, and that will tell us like what's the subset of people who benefit from antivirals like this, and what are their sort of biomarkers, so we can target the right people for the right therapy. But right now, I don't think we have enough insights to say, okay, you need to shut down this virus or you need to shut down this cytokine for treatment.
So here is a question. Can you explain the elevated antibodies? Are they less potent or effective because of the chronic activation, or were they less potent and effective to begin with?
Excellent question. I wish I had a time machine to go back and collect the samples from people before, during the acute phase. There are other studies, though, that have looked at the disease course of people who've gotten COVID during the acute phase. They measure the antibody levels, and they've seen a positive correlation with having elevated anti-nuclear antibody to a shorter disease course. So this makes immunological sense, because if you can mount a rapid, robust neutralizing or blocking antibody early during the phase of the infection, then you should be able to recover from the infection. Whereas if you started off with a poor level of antibody, poor level of T cells, you could have these lingering levels of virus, and that could lead to these chronic syndromes. So I believe, even though I have very little data to support it, that the failure to mount a robust immune response earlier in the phase of the infection may have led to long COVID. Yeah.
So there are about 50 more questions. I know time-wise we are running short.
We're getting over time.
Yeah, so do we respond to them later, or could a few questions be answered?
There's a couple of options. I think, that's a lot of questions. So I think it'll take a little while for you to kind of go through them. There probably are some duplicates, which might be helpful. What I would suggest is we go through them, and then this will be recorded, so we can add some slides to the end and put some answers to some of those questions. I can also say that, November 30th is the next town hall for Listen. So that's another opportunity for the Listen members. So if you aren't in Listen yet, you can do that. And then they have Akiko and Harlan do a monthly town hall meeting for members of the Listen study to get questions and answers and talk more specifically about the study, but also to do this type of dialogue. So that's happening on November 30th. So there'll be another opportunity to answer more questions. Akiko, thank you. This has been an incredible opportunity for people to get answers to their questions and to hear the amazing research that you're all doing. I loved personally seeing those the responses to our Kindred surveys, and to see what is coming from that, the information that's coming from that. So I encourage people that are on Kindred but haven't answered their surveys yet to answer those surveys and get them over to the Listen team. As you can see, that data is incredibly important. And so thank you to everyone who's participated so far. And I just want to call out Talia, who you see on the screen as well. She's our community manager on Kindred, and if you get emails from her, and she's our content person and is doing an incredible job on that end, keeping everybody up to date and informed. So thank you to everybody here.
Just saying thank you.
Yeah,
Great.
thank you, everyone. Really
Thank you.
appreciate your questions. Yeah, thank you.
Very much. And we'll hear from
I
you.
love all
again.
the hearts and claps and everything.
I
That's
know.
nice.
Thank you.
Screenshot that.
Wonderful.
Amazing.
Oh, thank you. Thank you.
Great.
See you next month. Wow. All right. Thank you.
Thank you all.
Bye-bye.
Bye.